Thanks for being here, and thanks to Stephanie for the invitation to speak. I'm a novice in this area, and I come here from a more basic science perspective, and so what I want to do is give you some background on the work that we're doing. I'm not going to share really data with you, but so we've been starting to work on sort of a next generation PZP vaccine a couple of years ago. Let's see, go forward. Yeah. John Turner, he has been mentioned a couple of times. So he's one of the pioneers with Jake Kirkpatrick on the use of PZP vaccine in wildlife management. So he came up with a number of criteria that for an ideal fertility control strategy, and I should mention directly that none of the fertility control strategies that's being used certainly meets all these criteria, but we're trying to improve and get there. So they should be, of course, highly effective, a multi-year duration, as Alan already mentioned, with the PZP-22 is a goal. Ideally reversible, although I guess you can question whether how necessary that is. Sorry, the cougars don't really meet that criterion, being a reversible control strategy. And then also no loss of basic social structure, no effect on the existing pregnancy, so they shouldn't cause abortions or stillbirth. And importantly, the application should be safe for people who administer prepare and administer the application. And then already mentioned shouldn't pass through the food chain, and then ideally delivered remotely. And an important criterion is that they should be cost effective. And you heard the cost of the PZP-22 that varies anywhere from $225 to $500. So that's getting up there in cost. Immunocontraception is one of the methods, and really this is about mobilizing the immune system to inhibit fertility. And there you've heard a number of targets have been mentioned. So GnRH, gonadotropin releasing hormone. There's a commercial product on the market made by Pfizer, now Zoetis, that is used in the swine industry to immunocastrate boars. But there's also Gonacon and for use in wildlife. There's an oocyte-specific immunocontraception that focuses on the Zona Pellucida proteins, and I'll focus on those. And then there's some research on trying to identify sperm-specific targets, but as far as I know, they haven't really been used in vaccines, at least not in any vaccines that are used in practice. So the Zona Pellucida vaccine is focused on the Zona Pellucida proteins, and there are a number of Zona Pellucida proteins as listed here. So there are 4 proteins. It depends on the species. So some species of animals have 3 Zona Pellucida proteins, but humans and horses, for example, have 4 Zona Pellucida proteins, and these are present on the outside of the membrane. So the Zona Pellucida envelopes the egg after it's released from the ovary after ovulation. The Zona Pellucida proteins have an important role in the binding of sperm, especially ZP3, and there may be some role for ZP4, and that then is important in the fertilization of the egg. The other protein than ZP2 is important to prevent polyspermy, meaning that multiple sperm cells can enter the egg, and that would interfere with fertilization. So the main targets that are being explored are ZP3, and it's pretty well understood that is probably the primary target. And then there's some research on ZP4 and ZP2 as targets for Zona Pellucida vaccine. So simplifying this a little bit further, so the goal is really to to prevent the binding of the sperm cells to the Zona Pellucida proteins by generating antibodies, and the antibodies bind to the Zona Pellucida proteins and prevent the sperm cells from binding to the Zona Pellucida proteins. So how do we do that? Well, so there's a number of steps that are involved in vaccines, and I just gave my final immunology exam to the vet students, and I'm not going to ask you to take an exam after the end of this lecture, so I will keep it simple. So after you inject an animal with a vaccine, what happens at the site of injection is a critical aspect or critical part in the immune response to the vaccine. But in the skin and in the muscle, there are cells which are a type of white blood cells. We call those dendritic cells, which act basically as sentinels. And so they sample the environment and continuously sample the environment. And so when you inject the vaccine is picked up by these dendritic cells, and then it's carried to the lymph node that drains the skin or the muscle. And there it activates other white blood cells, CD4 positive T cells and B cells, and then the B cells, with help from these T cells, differentiate into so-called plasma cells, which are basically factories of antibodies. And so they produce a lot of antibodies, an estimate of 1,000 antibodies per second, and they can do this for weeks or months. So they are enormously productive in producing antibodies. So this process takes some time, and so it takes about a week before we see the appearance of antibodies in the serum of an animal that is vaccinated. Okay, now we are going to go a little bit more into the vaccines, and so I gave you here a number of examples that you're probably then familiar with because you probably all received these vaccines. So there are different types of vaccines. You have a modified live vaccine. Basically, it's a virus that has been weakened, and so it does not cause disease, but it can still induce an immune response. So an example is the MMR vaccines, measles, mumps, and rubella. There are inactivated vaccines, such as the vaccine that was developed by Jonas Salk against polio. And then there are subunit vaccines, which consist of isolated proteins or sometimes polysaccharides. And really the PZP vaccine is modeled after these subunit vaccines. So they consist of purified proteins. And so these subunit vaccines, now in order to get an immune response, the proteins, even if they're bacterial or viral proteins, don't induce a robust immune response. And so you need to include a second component, and that's the adjuvant. And you've heard mention the use of adjuvants in the PZP vaccines. So the antigen is necessary because the antibody target the antigen. And the adjuvant is necessary to trigger the immune response and boost the immune response. Now, I have to mention a veterinarian who actually coined the term adjuvants. That's Gaston Ramon. He was a French veterinarian, graduated from the vet school near Paris. And after he graduated, he was employed by the Pasteur Institute to make antibodies against diphtheria and tetanus. And in those days, there were no vaccines against diphtheria and tetanus. And so they used to treat patients with diphtheria and tetanus with antibodies from horses. So they would inject humans, with antibodies against these diphtheria and tetanus. So he had a stable of horses. He would inject those horses with inactivated diphtheria and tetanus toxin. And then would collect serum from those horses and would partially purify that, and that would then be used to treat the human patients. What he observed is that the horses that developed abscesses at the injection site had a much higher antibody titer, a much higher level of antibodies than the horses that had no reaction. And he thought, well, that maybe I can, and that those abscesses initially were inadvertent induced because they didn't really purify the proteins very well. But then he thought, well, maybe I can induce sterile abscesses. And so what he did, he He combined the diphtheria and tetanus toxin with a variety of things. He tried breadcrumbs, he tried tapioca, and he tried metal shavings. And it all seemed to somewhat boost the immune response, but needless to say, none of these have actually been used commercially in human vaccines. But the methodology caught on, and we now use aluminum salts, and we use emulsions and other substances as adjuvants. So adjuvants are used to increase the level of the immune response, the magnitude of the immune response. They're induced sometimes to induce a specific type of immune response. So sometimes you need more of a cell-mediated immune response, sometimes it's more of an antibody-mediated immune response, and you can drive that difference with adjuvants. You can then also use it to decrease the antigen dose. Often the most expensive component of a vaccine is the antigen. And so if you can reduce the amount of antigen, that will make a vaccine cheaper. And you can also reduce the number of injections and then increase the duration of immunity. So by increasing the overall magnitude and duration of the immune response, you make a vaccine a lot more effective. Okay, so as you've heard, there are a number of PZP vaccines available. So the Zonastat H for horses and Zonastat D for deer, identical in composition. They're composed of a protein extract from swine ovaries. The first dose is given with a complete Freund's adjuvant, and the second dose with an incomplete Freund's adjuvant. And then, so they have been, as you heard from Alan, been approved by the EPA for use in wildlife. Induced about a year of infertility, and so that led then to the idea of making a more of an improved version of this vaccine, and that led to PZP-22. Again, the same type of protein extract, but now combined with a controlled release polymer. This is this polylactide glycolide, as well as a different adjuvant. QS21, it's a saponin, it's an extract from a South American bark tree that happens to have an adjuvant activity. And that induces at least 2 years of infertility when combined with this, essentially this primer, this modified complete Freund's adjuvant. And then Spayvac, you'll hear next from Mark about Spayvac, so I'm not going to talk more about that. So Zonastat and PZP-22, they are effective, but there are some problems with it. First of all, as I said, they're derived from porcine ovaries, and so the extract is fairly crude. It's not the purified ZP proteins, Zona Pellucida proteins, and so you may also get an immune response against other ovarian proteins, which may damage the ovaries, which may or may not be that critical. I mean, it depends on whether you want a reversible infertility or not. But it is a somewhat crude preparation, and there's some issues with the quality control of that. Also, the Freund's adjuvant is not an ideal adjuvant. It's first of all difficult to mix. It's a tough emulsion to mix. And also, it's actually a painful and adjuvant, and it can cause abscesses. And there have been more and more studies recently about abscesses in horses and donkeys that have been immunized with the Zonastat type of vaccine. And in fact, we can't use a Freund's adjuvant in laboratory animals. I mean, our IACA committees don't allow us to use those because of the pain that it causes. And then importantly for our purpose of making getting a longer lasting immune response is that this is not, these emulsions are not compatible with controlled release formulations, and you need a water-soluble adjuvant to achieve a controlled release formulation. And that's why in PZP-22 they used QS21, but that's not that stable, and the release profile from the pellets is not well known, and so it's not exactly clear how it actually works. So we are trying to come up with a next generation improvement. I mean, the good news, I should say, is that we know that this works, right? I mean, so the concept is valid and it certainly works, but there is room for improvement. And so what we're trying to accomplish now is to make an effective, long-lasting immune response, a vaccine that induces a long-lasting immune response after a single injection that is easier to administer, safe for animals and the environment, and then also well-characterized and consistent. And so the approach that we're taking is to come up with a better adjuvant that is potent, safe, and compatible again with the controlled release formulation. Develop recombinant proteins, and then combine that in a controlled release formulation. About the time that I was actually approached to see whether I could help and was interested in helping with trying to develop a next generation of this PZP, we had just come across this new adjuvant that is basically, I mean, I come from, I live in corn country, and so this is an adjuvant that is derived from sweet corn. And there's actually a lot of interest in developing new adjuvants, and there's a lot of interest in nanotechnology, making engineers can manufacture nanoparticles with all interesting kind of properties. But the difficulty is that they are often very expensive because the ingredients are expensive, the manufacturing process is expensive. So we stumbled across the fact that a variant of sweet corn, and this is a spontaneous mutation in sweet corn, that this variant of sweet corn makes nanoparticles. And these are nanoparticles derived, basically they're polymers of glucose molecules. So in normal plants, normal plants make starch, which is a linear polymer of glucose. And in the these sweet corn variants, they, instead of making a linear polymer of glucose molecules, they make these highly dendrimer forms of glucose molecules, and that results in the formation of these particles. So we tried a number of different surface coatings of these nanoparticles, and then came and tested them in an in vitro system. And then number 11 was the one that actually worked best in our hands. It's a surface modification with some simple chemicals, but with complex names, so that's why I just use the abbreviation here. And you can see these particles here. This is an EM picture, electron microscopy picture. They're sort of cauliflower-shaped, spherical particles, and then they're about 80 nanometers in diameter. And 80 nanometers, to give you a perspective, that's about the size of an influenza virus. And so this is sort of the size of a virus, so it's an ideal size of a particle to be taken up by white blood cells and be then presented to the immune system. And so we've demonstrated then that these nanoparticles work. They have an adjuvant effect. This is a study in pigs where the pigs were immunized with a protein. In this case, it's an egg white protein, ovalbumin, and then the egg white protein with nano 11. And this is an indication of the IgG antibody response to the ovalbumin. And we see that there's a robust induction of antibodies. Now, for this purpose, to make an effective and a long-lasting immune response, we think that the nano 11 particles themselves are probably not sufficient. And so what we want to do is what we're in the process of doing is combining nano 11 with other molecules that boost these dendritic cells, which, as I mentioned, are so critical in initiating the immune response, to sort of activate them from different sides and to get maximum activation of these dendritic cells. And so we do that in a system where we are doing it right now in a study in horses, and we're doing a series of in vitro studies where we take a blood sample from the horse, we isolate the white blood cells, and then test them in vitro in test tubes, essentially, or in a microtiter plate, with different combinations of the nano 11 with different other immunostimulatory molecules that we know have an immunostimulatory effect, and then looking at which combination works best. And we are about to do a number of trials in horses. Actually, on Friday this week, we're initiating a trial and seeing whether how well these combinations actually work. And this shows some of these combinations that we're using, and this particular combination where you combine all three of them, the IL-1 and TNF and CXCL1. These are just indicators of activation of the immune system, work pretty well. So we're pretty encouraged by this. Now, the other aspect of this next generation vaccine is to make recombinant porcine Zona Pellucida proteins. And so we have expressed, what you do is basically take the genes that encode for these proteins, put them in a plasmid, so a DNA that is then inserted into a bacteria, and then you can grow up the bacteria and isolate the supernatant, so they secrete these proteins. And then you can detect these proteins in the supernatant. So I'm not going into the technical details, but we have good constructs now for the expression of ZP3 and ZP2 and ZP4. And so in the pig, those are the three proteins that are expressed. So the pig doesn't have CP1, and the horse does have CP1, and humans have CP1. But the role of CP1 is probably minimal in the fertilization process. So we think that with ZP2, CP3, and ZP4 that we have a good targets to use for this next generation PZP vaccine. So with that, the people in my lab that are working on this, postdoc Jungi Mosley, and then technician Anissa Dunham and Fang Jia Lu. These are Laurent Coutil is an equine clinician, and Carlo Lav is a resident, equine resident, and they help me with getting the blood from the horses and immunizing the horses. And Keenan Park is a professor in biomedical engineering, and once we have identified the right combination of the adjuvant and the Zona Pellucida proteins, he's an expert on controlled release formulations, and so we will work with him to develop a controlled release formulation. And then John Turner I've been talking with him on a regular basis, and he's provided some of the PZP, the crude sort of native, I should say, native PZP for some of the experimental studies. And we've received support from the U.S. Department of the Interior and the Bureau of Land Management and the Baker's Trust for our studies. So, thank you.